All right, thank you so much for the invitation to present. And I'll be talking today primarily about the staphylococcal members of the skin microbiome. And so, first, starting out with a little bit of introduction. So, my group at the Jackson Laboratory is a hybrid computational experimental group, and we use a combination of microbial genomics, so i.e., the examination of microbes isolated from mixed microbial communities, which we then look at with very high precision one by one. And then we contrast this with shotgun metagenomics, which is an approach that studies the collective bacterial, fungal, and viral genomes in a microbial community. And to do so, we deconvolute this kind of high throughput sequencing data with a variety of computational tools. And most of these work by creating different reference databases of genomes of different species or genes of interest, and then mapping those short sequence reads back to those reference databases to figure out what these reads belong to. And so we've employed these approaches to reconstruct the microbiome of the human skin, which is a very complex ecology where the different skin sites can be very different in their physiology, with different amounts of oil, moisture, glands, hair follicles, and so on. And so here I'm showing the major environments of the skin. So oily and blue, like our face and our torso, moist and green, like our feet, or dry and blue, like our hands and our forearms. And one of the very striking things about the healthy skin microbiome is its strong skin site specificity, where the composition and the function of the microbial communities are driven by those physiologic characteristics of the skin. So what you're looking at here are relative abundance pie charts, and the different colors here represent different species. And you can see this site-specific diversity just by skimming by eye here. And in particular, you can see that the staphylococci in blue here is a ubiquitous and dominant player at most skin sites. And I mentioned this because this will be a major player in what I'll tell you about today. Now, these staphylococci, so they happen to not only be ubiquitous in healthy skin, but they're also ubiquitous as well as dominant in skin disease. And so I'll give a few case studies here. So this first one, which I conducted as a postdoc at the NIH, we looked at the microbiome of atopic dermatitis, or eczema, which is one of these inflammatory skin conditions. The hallmark of this skin disease is that patients lose the natural biodiversity that's typical of healthy skin. And this is shown by the relative abundance bar plot on the left, with lots of colors showing many different microbes. And on the right is the disease microbiome, showing a significant increase in the relative abundance of a canonical skin pathogen called Staphylococcus aureus in the blue. In our second case study, we looked at the changes in the microbiome in congenital ichthyosis, which represents a group of monogenic mutations that result in different biochemical changes in the skin, manifesting as scaliness, erythema, and skin infection. However, in this particular case, we were really surprised to see, despite the obvious skin barrier breaches and changes, we didn't see enrichment of that classical pathogen Staph aureus. In fact, it was almost nowhere to be seen. And what we saw instead was a shift in the non-aureus staphylococci. And so here we're looking at the staphylococcal composition of the skin microbiome at 10 different skin sites on the x-axis of an ichthyosis patient versus a healthy control. And as you can see, hominis and epidermidis species, so purple and yellow, are typical of healthy skin in contrast to the predominance of Staphylococcus capitis in the dark blue in the ichthyosis. In our third case study, so we looked at the skin, oral, and gut microbiota across lifespan. And what we observed was that the most dramatic changes in the microbiome within the skin of these frail older adults. And so this is the rightmost plots here on the edge. And so these are individuals who had the most dramatic health deficits resulting from their aging. And so here we're bringing in now those different skin physiologic categories here. So dry, moist, foot, and then oily sides. What we saw was that the dramatic remodeling of the skin microbiome was driven by depletion of Cutibacterium acnes in the red, concomitant with expansion of multiple staphylococcal species in blue. And again here, not necessarily those canonical pathogens, but a number of lesser known species, particularly Staphylococcus petencophorae, as well as nosocomial variants of Staphylococcus epidermidis and others. And so from these broad surveys of the skin microbiome, you can see here that there's extensive variety of staphylococcal species that inhabit healthy as well as diseased human skin. And so the big question is then, what are all these different staphylococcal species contributing to the skin, vis-a-vis their effects on skin barrier, immune education, contribution to disease, and then others, and how can we then untangle this complexity? However, before we can approach this, there's numerous complications. And one of these a particularly important consideration is that in addition to the diversity of those different species, including uncharacterized genetic space, there's also a tremendous amount of genetic diversity that's encoded at the subspecies or what's called the strain level. So most of you are probably familiar with the concept of strains, but for those of you who typically study, say, a favorite model organism, I'll give you my favorite example of Brassica oleracea. So these vegetables are all the same species, but strain differences at the genetic level result in dramatic phenotypic differences. And from a microbial perspective, we care about strains because in many cases, diversity at the strain level drives disease. So a single toxin gene in E. coli can turn our cloning workhorse into a pathogen that causes kidney failure. A single nucleotide mutation can make a pathogen resistant to colistin, a last resort antibiotic. And depending on which strain you have, so green or red, because we all have E. coli in our gut, your disease susceptibility, severity, or treatment response can be very different. And we hypothesize that which strain you have is a major driver of different diseases. But it gets one layer more complicated. Do we acquire and maintain just one strain of a species within an individual, so all red or all green? Or do we harbor mixtures of different strains, which changes that picture of disease risk? Because even healthy people might then be reservoirs for pathogens or antibiotic resistance genes, or different strains might compete against each other. Location in the body might even be important. If you have a pathogenic strain in your gut versus your skin, would you be more at risk for different infections? And so we thought that understanding this very fundamental question was a key first step, whether we have one or many strains for a given species, and how do we acquire them and what happens to them during their stay in our bodies. And so the way that we looked at strain diversity was to cultivate microbes from the skin and then sequence their genomes. So the microbial isolate genomics route, which you recall from my very first slide. And the reason we can't do this with metagenomic sequencing is because metagenomics doesn't have enough accuracy to resolve very closely related genomes. And in fact, algorithms to infer strains tend to vastly underestimate strain diversity. And so we built a large, laborious library of isolates for our deep investigation of strain diversity, doing this in Staphylococcus epidermidis, which is one of those keystone commensal staphylococci in our skin. We all have it, and it's everywhere on our skin. While it's essential in immune education and other roles, though, it tends to be low in abundance, and that makes it very difficult to investigate with metagenomic approaches. On the flip side, it's also the most common cause of bloodstream infections and has been implicated in wound healing and eczema severity. And so understanding how certain strains are associated with disease will provide insights into its potential transition from being beneficial to pathogenic and potentially provide new treatment strategies that target those processes. So we cultivated and sequenced about 1,500 staph epidermidis isolates from 5 healthy individuals across different skin sites and compared their genomes to answer our basic questions. Just recapping them here. So to answer this first question, the first extraordinary result was that basically pretty much every isolate that we sequenced from within an individual was a different genetically distinct strain. And so I'm showing here a heat map of gene prevalences of represented individuals' set of strains. So this isn't gene transcription, this is gene content differences for a strain. So skin sites on the x-axis, gene prevalences are hierarchically clustered. And so first off, the heterogeneity you see in the gene prevalences reflects that strains within a skin site can have quite different combinations of genes. Again, underscoring that pretty much every strain is unique. The gene contents can be quite unique to the skin site. So like the gene clusters characteristic of the belly button, the umbilicus, as well as foot strains, suggesting a strong skin site specialization, perhaps due to a distinct skin physiology, that results in genetic specialization. But again, the overall heterogeneity everywhere else suggests that there are additional factors causing genetic diversification. So what could be factors that contribute to that? So maybe where these different strains come from. These strains are in fact so diverse that it's really unlikely for all these strains to have diverged from a single parent strain. And so we went back to think about the origins of these strains. So intuitively, it's that we get our microbiome from our moms, but then subsequent forces like transmission events might bring new strains in, or evolution might refine the gene content of the strains that we get. And so we built a phylogenetic tree of all of our strains, and then we use single nucleotide polymorphisms to infer the source of the strain and to examine the relatedness of strains within individual, because SNPs can act like a molecular clock for how distant two strains are, which is reflected by the closeness and the length of the branches on this tree. What we found was that, to our surprise, we're colonized by multiple founder strains. So, for example, P0 in yellow might get two strains from two different branches, P3 in red could get two strains from less related branches, and so on. And that's surprising because the first study within individual strain diversity from Eric Alm's group at MIT, looking at Bacteroides fragilis in the gut, showed that in their study that there was a single founder. So each individual had just one branch for its parent strain, which then successively diverges over time by accumulating mutations. Okay, so we acquired diversity via the pool of strains that found our microbiome. But what are other factors that could promote genetic diversification subsequently? Transmission between body sites with an individual is intuitive. So again, by tracking SNPs, we can map the most common ways for strains to travel, which is by touch. And so the lines here show the most prevalent likely transmission routes, with the darker lines, like those from the hands to the face, reflecting the more likely transmission events. And not only can strains move from skin site to skin site, but genes via horizontal gene transfer as well, which typically occur by bacteriophages or by plasmids. For example, a particular example of clinical interest is the occurrence and transfer of plasmids encoding antibiotic resistance genes, which we computationally predicted and verified experimentally, looking for evidence of dissemination of these genetic elements. Left, just an example of a plasmid predicted to bear several antibiotic resistance gene determinants. And on the right, we see a map of its dissemination with an individual. So the darkness of the blue dots reflect the prevalence of those genes in the strains at these skin sites. And so what we saw was that these resistance genes disseminated to multiple different strains at distant sites. Okay, so horizontal gene transfer can disseminate genes within an entire population of strains. And so we have an emerging portrait of strain diversity, where we have this extraordinary within individual diversity that's shaped by multiple evolutionary forces that starts with the colonization of a genetically diverse pool of founder strains that's then subsequently diversified by functional specialization to a skin niche, transmission of strains between skin sites, and genetic exchange between those strains. So now taking a step back, now the picture is a lot more complicated. So all this uncharacterized biodiversity now encoded not only at the species, but also at the strain level. And I won't go into detail here, but now that we've sequenced and analyzed about 5,000 more staphylococcal isolates across 24 staph species, we observe similar diversity, genetic diversity at the strain level for most of the other staph species as well. And so, what then we would ask is, what's the function of all of those strain-specific genes? And so if we go back to this figure where we see that tremendous gene content differences between strains, from an annotation point of view, so predicting cog functions here, so core genes, those shared by all strains at the top, and then strain-specific genes at the bottom, here for the umbilicus, toe web, and then everyone else, it was clear that the vast majority of strain-specific genes have no known function. And so we built a platform leveraging CRISPR interference for targeted knockdown of genes. And so this knockdown approach, where the catalytically dead Cas9 inhibits transcription by steric hindrance, we took this approach over others like gene knockout or transposon mutagenesis, because we didn't want to rely on efficient homologous recombination, which is often very difficult in primary isolates, or the use of transposons requires generating many mutants to cover the full genome. And we have a ton of different strains that we wanted to look at. And so the way this works is that we synthesize a comprehensive guide RNA library against all compatible sites in the genome or tile them across the genome, and then transform this into Staph epidermidis, creating a genome-wide knockdown library. We can then induce the knockdown phenotypes and then screen this pool in different environmental conditions. And then the way that we identify genes that have a growth defect in that condition is by amplifying the guide RNA and then sequencing it, basically using it as a barcode to quantitate the growth defect that results from each knockdown. And so this is a dummy example. So strains with knockdowns in a genes essential for growth in a condition will grow slower and become underrepresented in the pool, like this red strain. Resistant strains will grow faster and become overrepresented, like strain blue. And then by comparing log to fold change from the initial pool to the final as a proxy for growth rate, we can infer gene essentiality in a condition. And so what you have at the end of this assay then are candidate genes that are important for growth in a particular condition. And then to test for functional relationships, you can return to the original knockdown strains to perform follow-up studies. I'll give an example of the type of data we derive from this screen. So this is a volcano plot with adjusted p-values on the y-axis, log2 fold change on the x-axis. We validated the growth defect first of essential genes in the red, so showing the robustness of this approach. Validated experimentally by creating individual knockdowns. And then you can see here the differences in growth rate, i.e. optical density over time, when the knockdown is induced, shown by the red versus the blue curve. We performed this CRISPR interference screen in 24 diverse conditions that's relevant to skin and infection, and these included multiple stressors, so different antibiotic, osmotic stressors, a variety of different nutritive conditions that we thought could approximate different factors in the skin and bloodstream. And then we matched this gene essentiality data with RNA-seq. And our goal integrating these two large-scale data sets was to identify different genetic pathways that were either essential or transcriptionally dynamic in these conditions to understand Staph epidermidis's phenotypic plasticity in our bodies. And so I won't go into any of the individual results here just for the sake of time. And so this is just showing a high-level summary of our findings, where we identified condition-specific responses and pinpointed some mechanisms particularly relevant to the low pH, urea-rich, salty conditions of the skin. And so this first study was gene function, but just in one strain. And so since then, we've been systematically probing the role of the accessory genome in that lifestyle plasticity for 30 of these genetically diverse Staph epidermidis strains. And so stay tuned for the next year or two for our look at that. So we talked about complementing genomics and functional genomics approaches to characterizing strain diversity. And so our next step is to look at strain phenotype. So vis-a-vis their function on human skin. What's the phenotypic consequence of all that genetic diversity in vivo? But again, coming back to our big problem, there's so many species, let alone strains, that we need methods that accommodate throughput. And so we previously published some of our efforts to phenotype microbes in human tissue culture immune activation assays. But while these are informative in terms of characterizing the potential breadth of immune activation of lots of very diverse microbes, these 2D human culture assays have a fatal flaw in that the translation back to in vivo complexity is limited. So, for example, they don't capture the inherently spatially constrained nature of microbial interactions in the skin. And so to complement these high throughput approaches, we developed a mid-throughput platform that combines genomics with metabolomics and organoid technologies. So this starts with using comparative genomics to pare down from our thousands of genomes to a couple hundred of the most genetically diverse strains. And here we've selected 6 of the dominant staph species in the skin. Genomes, which we then complemented with untargeted metabolomics to understand how genetic diversity then translates to metabolic diversity. And so just showing an example here of how indeed that genetic diversity at the strain level results in metabolomic diversity. We're looking at the Z-scores of 2 different Staph epidermidis strains, as well as a media control, and that's a TSB. And they're grouped by categories of interest here. So tryptophan metabolites, for example, that mediate skin barrier repair and different immune activation conditions. And you can see just by eye here, major differences between the 2 strains in a number of these different pathways. And so now zooming out, we can look at a PCA plot of the metabolome of all 180 strains, so colored here by species. And the distance between the points here reflects a similarity between the metabolomes of the strains. And so what we see here first is that variation is primarily explained by species, which makes sense, but there's extensive strain variation in the secreted microbial products. So more pathogenic isolates like Staphylococcus lugdunensis and Haemolyticus cluster together up at the top. Aureus in green is in a class of its own, so that's the most pathogenic, I would say, of the staphylococcal species. And then interestingly, our canonical commensal Staphylococcus epidermidis, these strains bifurcate into clusters that are either pathogen-like or commensal-like, which I'm showing here in the red circles. So now what we're doing is we're correlating gene presence absence at RNA-seq data and the metabolomes to basically link the genetic origins to try to annotate a lot more of these different metabolites. So then the next step here is to colonize these staphylococci, so our 180 staphylococci that we subselected from our genome analysis and our metabolome analysis. We then use these staphylococci to colonize a 3D skin model. And this is called reconstructed human epidermis. And this model possesses major features of healthy skin, including the dermis, the epidermis, and the stratum corneum. And this structuring allows us to retain that aspect of spatial structure that we know occurs in vivo in terms of the interaction of the skin microbiome with the skin cells. And so here, just on the left, is how we do this in terms of our 3D model. We infect these skin organoids with bacteria or a water control. And then following 24, 48, or 72 hours of colonization, our readouts are either dual microbial host RNA-seq, so we capture both sides of this interaction, single cell sequencing, and spatial transcriptomics to examine host response, retaining both the cell type specific as well as the spatial responses of the skin to staphylococcal colonization. So I'll give us just a snapshot of each of these data types, highlighting some of our findings to date. So first, look, no genomics talk is complete without an enormous heat map. So we look broadly at log2 fold change over vehicle control. So here strains are on the x-axis, and then a subset of immunity-related genes are here on the y. And what we saw was that transcription bifurcated like the metabolome. So for example, the pathogens here in the reds, we saw that a subset of Staph epidermidis strains also bifurcated in terms of their host impact with a group that was more commensal versus a group that was more pathogenic, with the more pathogenic-like strains highlighted here in the black box. A unique feature of that pathogen-like cluster of these otherwise traditional commensals, which we've called the C1 group, was upregulation of IL-23, which is a cytokine that can promote chronic skin inflammation and has other known roles in infection response, but is really very little studied in terms of its role as vis-a-vis being produced by keratinocytes in the skin. Pathway analysis breaks down other features of interest where we observe differential gene expression of these strain clusters. So particularly this SPRR family, which has diverse but also largely uncharacterized functions, which include antimicrobial response and functions in skin barrier maintenance. And so we got really interested in the kind of the pathogen specific downregulation of these genes versus kind of the I would say the upregulation by the C2 purely commensal like cluster versus the C1 more pathogenic like commensal cluster. And so now, what we can do is we can follow up on the host mechanisms by doing things like knocking out these genes and keratinocytes that are used to reconstruct the keratinocytes that are used to construct the 3D organoid models, and then therein we can reconstruct host mechanisms via CRISPR knockouts. We can also generate hypotheses in terms of what microbial functions might underlie this differential response. And so here we correlated the human gene expression with the microbial gene expression, again, pulled from the same RNA-seq data. So this is an example here on the left. And this allowed us to triangulate on particular microbial genes or unknown genes in this case that might be underlying these different strain-specific host responses. So here, this heat map is just showing that correlation with some of the significant associations starred here. And then what we can do with this is that take those hypotheses produced by this kind of analysis, and then use comparative genomics guided by this analysis to examine presence or absence of genes. So here, taking that binary IL-28 induction, we identified SDRF and 3 hypothetical genes that were really strongly preferentially present in the genomes of that C2 pathogen-like commensal cluster versus the C1 commensal cluster. And what we're doing now is using our CRISPR interference platform to knock down these microbial genes to examine if they're causative in this particular interaction. And so, as a final example of our outputs, we asked whether the microbial response was restricted to those uppermost cell layers. So, again, recalling that those are made up of the keratinocytes and then the lower layers in the dermis, which is comprised of fibroblasts in that particular 3D model and in our actual in vivo skin. So to look at these spatial effects and these cell type specific effects, we use single cell sequencing, which I won't go through here because it was pretty analogous to what we saw here with a newer technology called spatial transcriptomics. And spatial transcriptomics overlays gene activity in the tissue sample, which is what you see here, a basic H&E slide, to map where these transcriptional changes are occurring. And so here, each dot is kind of like a single cell. So it's a little bigger. This particular technology, it's a Visium platform, so it's a little bigger than a cell. But each of the colors reflects the cells that have transcriptional profiles that are statistically distinct from each other. And so naturally, what you see here is that the cells are falling into their layers, as we might hope, with the epidermis, with the different keratinocyte layers and the browns and the pinks, and then the dermal layers in orange, and then this is an ex vivo skin explant. And so we also have a lower dermis and adipose layer there at the bottom layer in the blue. So here now, when we colonize with Staphylococcus aureus, the USA 300 strain variant here, we see significant transcriptional changes in some, but not all of these epidermal layers, as shown now in the pink. And strikingly, we saw that the broadest change is in the fibroblast layer. Looking then at the differentially regulated genes between the layers, we saw that the inflammatory response in red occurs in the dermal, but not necessarily the keratinocyte layers, and that's coming with a concomitant downregulation in skin barrier genes, which are broadly characterized in the green there. And so this suggests that this response, which is in theory, which in theory is the priming that would then recruit additional innate and adaptive immune factors in vivo, it suggests that this response is actually mediated by fibroblasts, not keratinocytes. And this was surprising because this is not the most proximal layer of the skin to the microbes. And so what we figure in this particular case is that there's a couple different hypotheses. First, that the microbes could be signaling directly to the fibroblast, or that the microbes could be signaling earlier to the keratinocytes, which then produce a factor that then activates the signal in the fibroblast. And so what we need to do next is refine this model with time series data to understand cell-cell signaling, as well as temporal effects, in addition to those spatial effects. So just to summarize, so that gives you some examples of how we've progressed from using metagenomics to look at the remarkable species and strain level staphylococcal diversity in the skin, to probing host mechanisms, which are more multi-omics organoid framework, which in vision will ultimately allow us to model patient-specific microbes as well as host genetics in this model to further probe these unique interactions. And just by way of showing you kind of the next phase of that spatial transcriptomics, what we're now developing is dual host microbial spatial transcriptomics. And here we've worked this out for about half a dozen different skin bacteria. And so here you're seeing, again, an example of the H&E slides. And then in red, we've been able to map the individual cells of bacteria in each of these models that have been colonized with that corresponding bacteria or with a mix of different skin bacteria. So we're bringing now both the dual microbial host RNA-seq to now capturing that spatial interaction so that we can also understand the spatial interactions of the microbial response together with those cell type specific responses in the skin. So with that, I'll thank my fantastic group at the Jackson Laboratory in Farmington, Connecticut, which is 2 hours north of New York City, 2 hours south of Boston. Much of the work was conducted by Wei Zhou, Anita Voigt, Elizabeth Fleming, Royu Yang, and Morgan and Georgia in the lab. Thanking all the different students who have contributed to these projects over the years. And then, of course, far too many collaborators to thank, so I'll just mention those pertaining to what I discussed today. So Len Millstone and Nan Ring on our skin model work, Shizhao Li and Zhidao Yao on metabolomics, and Tarek Oswalad on our bioprinted skin models, and Julie Robeson and George Kushel on our aging work. And with that, let me return to the chat and I can begin to answer questions.